This week we're covering trace and ultra-trace minerals, also known as microminerals. And your textbook and learning unit has a variety of information about these different minerals, so I'm not going to go into individual detail about all of those, but I wanted to kind of go over some common themes or characteristics that are similar among minerals. So as you're going through the chapters and the unit, that you can kind of think about how these sort of fit together or things that are common among minerals. So I'm just going to go through some general, again, the specifics are in the unit, but hopefully this will help get some of those general ideas. So the minerals that we're going to be covering this week are iron, zinc, copper, selenium, iodine, and molybdenum. So one of the common sort of themes or characteristics is that most minerals can exist in multiple forms or states. So specifically multiple ionic states. So this is that whole idea of oxidation reduction reactions. So we covered in an earlier unit redox reactions, and if you need a little refresher, I would go back to that because that's really a key factor with minerals, because many of them exist in different states. So iron is an example we used that exists in the 2+ state, which is ferric, or the 3+ state, which is ferrous. And so copper, zinc are also, they can exist in different states. So that allows those minerals to donate or accept electrons, which has a variety of implications. One of the implications is that that they can function often as antioxidants. So again, we reviewed previously what an antioxidant does in terms of oxidation and reduction, so feel free to go back and review that. The multiple states also sometimes affect how other nutrients interact with that mineral. So again, iron is an example with vitamin C, and we used this in the previous unit in terms of how vitamin C and iron interact to trade, essentially sort of trade electrons. And with this, these multiple ionic states that the minerals can exist in, that often affects how active they are. So often one form is maybe a more active state, perhaps one form is more absorbable, perhaps one form has specific functionality. So again, as you're going through the different minerals, think about what the different forms mean. So when that mineral is in a 2+ versus a 3+ or a 1+ versus a 2+ ionic form, what that means in terms of functionality. Another characteristic or things to consider when you're reading about the individual minerals is factors that affect absorption. So we already talked about those various ionic states that can affect absorption, so that's one factor. Another big factor is that many minerals have competitive absorption or they compete for the binding sites for transport. So an example here would be DMT1 is a transporter that transports iron, is what's shown here, and that's what it's primarily known for. It's a big transporter of iron. So here's your little iron molecule, and the DMT1 allows it to enter the enterocyte. So these little squigglies you generally know on the cells when it's like that, it's an enterocyte. So that allows iron in. Well, DMT also transports other minerals like zinc and copper to an extent, and some others. So the issue here is that in sort of normal states, when there's normal amounts of the minerals in the body, this all works well. But if there is an overload of one particular nutrient, that can sort of clog up, so to speak, the transporter so that it's not available for other transport. So for example, if we have someone who is on a high dose iron supplement, that may really bombard these transporters, and it may decrease zinc absorption to some extent. So it's just something to think about when there's a large amount of one mineral being provided, whether that might provide a lot of competition for the transport for other minerals to get in. Another thing to consider here with some of these important transporters when you think in terms of genetics is that if there is some sort of genetic mutation for the code for this transporter. So remember, every proteins are transporters, and every protein has a gene that codes for it. And so if that gene has a mutation that causes it to code a little bit off for the transporter, so maybe the transporter's structure is slightly different and it doesn't transport quite right because maybe the iron or the zinc or whatever doesn't bind exactly properly, that could decrease the person's ability to absorb that nutrient. So those are some things to think about in terms of competitive binding of transporters. Another factor that's common with minerals is chelation. So basically, chelation just means the binding together of two things. So I decided to put hand holding here because it's sort of more interesting sometimes than looking at minerals. So, but when we think about chelation, just that binding together, and with minerals, that binding can either be a good thing or a bad thing. So sometimes that chelation, if the mineral is chelated to something particular, it allows more or better transport or absorption. So maybe what it's bound to allows it to be moved where it needs to move. On the flip side, sometimes when it's chelated to something else, that prevents absorption. So chelation isn't good or bad, but it's something to be aware of when you think about minerals. Is it bound to something that's either going to allow for its absorption or in transport, or is going to prevent it? Another factor kind of in this realm, not chelation so much, but general absorption factors, is the pH of the environment can often affect absorption. So for example, zinc, chromium, and some others are generally best absorbed in an acidic environment. So when we think about our stomach and the upper part of our small intestine, that is a really acidic environment. However, if someone, for example, is on antacids, so they're taking antacids very regularly, that decreases the acidity of the environment, so it may decrease the absorption of those nutrients. So those are kind of all some factors that kind of are something to think about in terms of absorption. There's another variety of factors that affect availability of the nutrient, sort of I guess before it even gets into the person's body. So one of those factors is soil quality. And I think it's easy to forget this, and even when we, for example, if you plug foods into a nutrient database to analyze how much of a particular mineral is being provided, that all is kind of assuming a standard amount based on soil conditions, etc. But some nutrients, some minerals can vary, the content in food can vary widely depending on how rich the soil is in that particular mineral. So of course for plant products, this is particularly important. So it's something to think about, for example, when we talk about environmental issues like over farming, things like that. Soil quality is really important because it impacts what nutrients can actually grow into the food, because if it's just not available in the soil, it isn't going to get into the food. So just kind of, I guess there, I think it's important to recognize that even if we put foods into an analysis program and it says the nutrients are adequate, it's not always going to be 100%. There is, still a possibility that it's lower than we think. Another factor is food processing. So things like cooking can affect minerals. So heat doesn't destroy minerals like it can for some vitamins, but it can affect what the mineral is bound to. And so again, as we just talked about, that can affect absorption in some cases. So this is one of those things that in normal situations, normal cooking, all of that sort of thing, it's not going to be a huge factor, but it's just something to consider as you think about the characteristics of minerals. Along with that, sometimes the mineral actually has to be released from the food that it's bound to in nature, and so cooking helps make it more available. So it doesn't just go one way or the other, but just something to consider. So sort of food processing and preparation and how that affects the minerals we actually get into our body from the diet. Another sort of general category of sort of characteristics, I guess, of microminerals is they often are part of some other kind of complex. So there's a few different ways that this can happen. One is that it can be a cofactor for enzymes, and that's very common, that the mineral is a cofactor for enzymes, meaning it binds to the enzyme and allows the enzyme to work. So one example that I have here is superoxide dismutase, or SOD. This is a very powerful antioxidant in the body, and this is a structure of it here. And you'll see in the middle of this enzyme is copper and zinc. There are little sort of pockets for that copper and zinc to attach in that enzyme, and the enzyme is only active when it attaches. So you can kind of see here how two minerals are necessary for this enzyme, superoxide dismutase, to function properly. So in terms of the function of superoxide dismutase, over here we have excuse me, we have the superoxide free radical. So this little dot here that you see indicates that it's a free radical. So this is a really common free radical in the body, but it can cause cellular damage. And so superoxide dismutase can facilitate the conversion to hydrogen peroxide, which is not so harmful, and oxygen. So again, this is a really powerful and common antioxidant in the body that relies on two minerals for it to function properly. Oops. And I forgot one other piece I wanted to put there. It can also, not related to the specific example I have here, but in terms of being part of another complex, sometimes it's not as a coenzyme, but it's actually just part of the structure. So, for example, copper is a part of ceruloplasmin, which is a glycoprotein. And because copper can change ionic states, how we just talked about, so copper goes between the 2+ and the 1+ state, it allows this compound to oxidize other minerals, but in a positive way. So sometimes we think about oxidation as being bad, but in a positive way in this case. So this general category that these are can be part of other complexes, either they can be something like a cofactor that it attaches when an enzyme or something needs to be activated, or it can be just part of the sort of core structure of another compound. Okay, now we'll move to the next one, and this is the last general category. So multiple minerals can act in some way on transcription. So just kind of remembering that transcription is really the first step in making a protein. So when we make a protein, the DNA, there's a gene that will code for the protein, and the first step is copying the code in the the code in the DNA to messenger RNA, so that that can go through the process to gather up the amino acids to make that particular protein. So with transcription being the first step, we need transcription factors attached to the DNA to allow that transcription to start. So there are a variety of minerals that can either be part of the transcription factor itself, or they might bind to the transcription factor to stimulate transcription. So minerals can be very involved in gene expression. So again, to kind of back out and think of this as a big picture, what I really hope this helps you do is kind of think about some of those core themes. So as you're going through the materials for this week, not the minerals for this week, but the minerals in the materials, as you're going through all that for this week, you really think about sort of those common themes to help you just kind of organize the information. And then of course each of the minerals has some very specific functions that you can look at as well.